Hello, very good day, dear practitioners. Welcome to this interesting and special webinar we have here today. Today, we have the presence of Jacques Bower. He is a pharmacist, graduated from the University of Lausanne, is a clinical trainer, and is the founder and co-owner of the AWD laboratory. He is the ministerial delegate for preventive health and well-being in the population of Savoi. Therefore, it is a great pleasure to have him here with us, and we are going to talk about what biophysics is, a new concept in biotechnology. So, welcome, Jacques. It is a pleasure to have you here for all of us, and an honor. Good morning, everyone. It is truly an honor for me to co-present this seminar, especially alongside Esteban. Good morning, Esteban. Good morning, Jacques. I apologize for not speaking Spanish, although I understand it. That's why I will make my presentation in English. Thank you for your understanding. Perfect. So I think without further ado, we should just get started. So welcome, Jacques. I'll let you take the spotlight, and it's going to be a real pleasure to listen to you.
Good morning, and thank you very much for being there. What I would like to share today is not simply a technology, but a way to understanding living systems through the lens of biophysics, epigenetics, and functional interaction. For a long time, biology has been approached mainly through structure, measurements, and isolated parameters. But living systems are dynamic. They are interconnected and constantly adapting to their environments. This is why I believe we need to complement quantitative analysis with a more qualitative and integrative perspective. In this webinar, we will explore how biological signals, epigenetic modulators, and technological tools can help us better understand functional imbalances, adaptation, and the possibilities for optimization. My intention today is not to oppose conventional science and emerging approaches, but to build a bridge between them in a rigorous, practical, and clinically meaningful way. Indeed, the most insane way to approach science is to dig trenches where common sense actually calls for building bridges between disciplines. To summarize, your body is not a collection of separate parts. It is a living network of relationship. So I would like to give you just a couple of information regarding this laboratory that I created lately in the midst of 2021, exactly during the period of the COVID. And this laboratory appeared to me very important in order to really understand something more than usually people had in mind when they were talking about immune system, infection, but also what we call the bioresonance or quantum biology approach, which is more qualitative than quantitative.
Very interesting, because it is somewhat the process, Jacques, of how we integrate what are the quantitative technologies and correspond them with the qualitative ones. So go ahead, it will be very interesting to hear you.
Thank you very much, Esteban. Here's a little picture of the laboratory, and you can see two clients or two patients who are just right over there. And this laboratory was very important because there were a couple of technologies involved, like one where we are really among the best in the world, which is live blood analysis through a very specific microscope called dark field microscopy. But we've got also a detoxification strategy, which is called Firewalker, because you warm up the feet of the patients. The Firewalker detox technology is made out of heavy metal and toxin elimination support as a basis, but there are many other things that we can also eliminate and check exactly the content of what is eliminated after patient's treatment session. And the last one is called NLS for nonlinear system technologies. And this is something very important also that we will discuss in order to understand that nonlinear systems means that in a certain way, you don't have the same mathematical results by doubling a dose, for example, but sometimes just a little impulsion can create some enormous differences by biological human systems. And we will come back to that later on. We have other technologies which are involved in this laboratory. One which is called SHEVA, cryotherapy, which is a kind of cellular regeneration and anti-aging supports. And SHEVA means simply heat extract weight activation. And I've been cooperating with a professor from Cambodian origin in Paris who found a way which is called, it's not exactly a cryotherapy, but this is a way to threaten the fatty cells to die if they don't transform immediately their content in fat into energy. And that works very well. And this is non-invasive. And this is just one of the things that we are doing as well, because it goes also through epigenetic modulation. I will come later on that, through the activation of a protein which is called histone. I continue now with the epigenetic modulator optimization reports. And that's exactly the reason why I'm in front of you today, after I could explore and validate a methodology which is provided by the company Epixlife. And that's the reason why I was so honored to create this webinar together with Esteban today. We've talked a lot of time together, and we decided that it was time to make this webinar because epigenetic is something absolutely important, I would even say essential, that cannot be left to sides today, even in the context of clinical and university medicine. And by the way, I would like also to add something which seems to me quite important. My interest for genetics is not recent. You can see here a very important and interesting historical picture of myself, a bit younger, next to Dr. James Watson, who is the Nobel Prize for his discovery of DNA double helix structures in 1962. And that was really for me an extraordinary encounter. And I used to be invited, that was in 2013, in this laboratory in New York, next to New York, in order to cooperate with them. And that was, of course, much more intended towards genetics purely, but that was the basis and the background of my growing interest for everything around genetic, including, of course, epigenetics later on. And now let's come to the conceptual foundations. We are talking about biophysics of complex biological systems. And in order to understand that clearly, we have, first of all, to make a clear distinction between genetics and epigenetics. I have to talk about the Human Genome Project that you can see here on the left side of the slide. The definition of the Human Genome Project is an international scientific project that mapped the entire human genome. This was the moment when science tried to read the full book of the human body. When we talk about genotype, the definition is, of course, the complete sets of genes inherited by an individual. And the genotype is, in certain manner, the blueprints, like doors already installed in a house. You cannot remove them so easily. But there is something that you can do with the doors. I will come that later on this. And as regards DNA testing, the definition is the analysis of genetic material to identify biological predispositions and variations. We look at what is written in the code, but not necessarily how it is used. And now we are coming to the right side of this slide, which is, of course, the most important and which is the core of what we are discussing in this webinar today, which is epigenetics. So first of all, you have to understand that epi comes from the Greek root, which means above. So you have the genetic on one side, and you have the genetic above the genetic on the other side. So what is epigenetics exactly? Epigenetics studies how gene expression is regulated without changing the DNA itself. In other terms, you still have the doors in the house, and they are very difficult to remove. But you can choose the way the door is open or closed or half open, right? And that's where you have the possibility to intervene into this field called epigenetics. And it's not about what is written, like on DNA substance, but it's more about how it is read and expressed. Now, we are talking about the phenotype, and the phenotype comes from the Greek roots phenine, which means how does it appear, which is very important. So the definition of phenotype is the observable results of a gene expression, how genes actually manifest in the body. And you will see later on that there is a new science called nutrigenomics, which shows how what you eat can influence whether your door will be half closed or completely open, and so on and so forth. That's quite interesting. And now, as regards the phenotype, I would say this is really what becomes visible, what is expressed. As regards the external modulators now, the definition is the environmental factors that influence the gene expression. For example, you have the nutrition, you have the stress, the toxins, the sleeping patterns, the emotions. These are all the external factors that influence how our genes are expressed. Now let's see at the third point of the right part of the slide, which is epigenetic modulator optimization reports. The definition clearly is reports that identify which factors can be optimized to improve the gene expression and overall balance. These tools help prioritize what can be adjusted to improve the body's functional balance. In other terms, genetics gives us the potential. Epigenetic determines how that potential is expressed. In other terms, if I can say so, genes load the gun, environment pulls the trigger. In other terms, the genome is not a fixed destiny. It's not a prison, but a modifiable potential. Now, in order to understand clearly what are the complex biological systems, we have to differentiate three things. What we do call interconnected systems, cellular communication networks, and nonlinear systems. As regards the interconnected systems, their definition is a biological systems do not function in isolation. Organs, tissues, and systems are constantly interacting with each other. The body is not a collection of independent parts. It is a network of constantly interacting systems. In other terms, nothing in the body works alone. Everything is connected. Now let's talk about the cellular communication networks. The definition is cells communicate continuously through biochemical and bioelectrical signals to maintain balance and coordination. Cells are constantly exchanging information, days and nights, through chemical signals electrical signals, and complex communication pathways, including light. And there is a wonderful scientist called Fritz Albert Popp who has shown this. So the body functions because cells are constantly talking to each other. Now let's come to the nonlinear systems. In biological systems, cause and effects are not always proportional. Small changes, as I said before, can produce large effects and vice versa. This is also what we call the butterfly wing effect. In living systems, the response is not always predictable. A small input can create a large effect, and sometimes a large input creates almost no effect. In other terms, the body does not behave like a machine. It behaves like a complex adaptive system. In conclusion of this slide, I would like to say that when we understand these three principles, interconnection, communication, and nonlinearity, we begin to understand why a purely reductionist approach is often insufficient. Now, let's come to the qualitative approach that we want to oppose to the quantitative approach. The qualitative approach, at this point, we move from a purely descriptive view of biology to a more functional and integrative perspective. As regards the relationship between epigenomes and environments, the epigenome acts as an interface between genetic potential and environmental influences. The epigenomes is where the interaction happens between what we are genetically programmed for and the environments we are exposed to. Your genes provide the potential. Your environment shapes how that potential is expressed. In other terms, we are not only the results of our genes, we are the results of how our environment interacts with them. Now, let's look at the right part of the slides, qualitative versus quantitative perspective. First of all, let's understand clearly what we are talking about. The quantitative approach are the measurable values like number, concentrations, levels. As regard the qualitative approach, it's more like interpreting the patterns, the relationships, and the functional states. A quantitative approach tells us how much. A qualitative approach helps us understanding how and why. Numbers are important, but they don't always explain the whole picture, especially in complex biological systems. In other terms, we don't just want to measure the system. We want to understand how it behaves. In complex biological systems, understanding relationships and patterns is often more informative than looking at isolated numerical values. And I allowed myself to add slides that I find especially interesting to illustrate that difference, which is this one. As you can see, this scientist is looking right at the bottom of the glass, seeing how the water in the glass behaves, and is completely forgetting about the fact that a tsunami is just about to overflowing. And that's exactly the difference between building bridges between different disciplines and ways of interacting with the matter and the biological systems, and the way to not do it. So why can a qualitative approach be more relevant than a purely quantitative one in complex biological systems? There are a lot of reasons for that. But what I would like to say is that, of course, the in many areas of science, numbers are essential. They give us a precision, measurements, and comparison. But in complex biological systems, numbers alone are not enough, because living systems are dynamic, interconnected, and non-linear, as I was saying previously. A purely quantitative approach tells us what is happening, but not always how or why it is happening. A qualitative approach allows us to understand patterns, relationships, and functional states within the system. In other terms, in complex systems, understanding relationships is more often informative than measuring isolated values. Two patients can have the same numerical values in the test, but completely different functional states. And this is exactly why we need to integrate qualitative approaches into modern biological analysis. In other terms, and to make it short and clear, numbers measure, but relationships explain. Now that we have explored the fundamental concepts, let's move to how these principles can be applied in a biological and technological framework. What we are talking about here is the integration of biological understanding with technological tools. In other terms, we are connecting biology with the technology. Biophysics provides a bridge. I was talking about bridges previously. It allows us to translate complex biological processes into measurable and interpretable signals. In other terms, biophysics is the language that allows technology to interact with the living systems. And instead of looking only at static structures, we are now looking at dynamic interactions, energy, signals, and communication within the biological systems. In other terms, we are no longer just analyzing the body. We are analyzing how it functions and communicates. This approach is consistent with what we discussed earlier: interconnected systems, cellular communications, and non-linear behavior. If biology is dynamic and interconnected, then technology must be able to capture that complexity. And this is exactly what modern biophysical technologies aim to do. In other terms, and to summarize it in one sentence, biophysics allows us to translate biology into measurable signals and technology into meaningful insights. Now, let's talk about them. Let's start with something very simple. An electromagnetic signal is a form of energy that carries information and travels as a wave. In simple terms, it is how information moves through space. We use electromagnetic signals every day, for example, with Wi-Fi, mobile phones, remote controls, etc., etc. So the question is not whether electromagnetic signals exist. We all know that they exist. But whether biological systems also use this type of communication. And the answer is yes. Biological systems generate and respond to electromagnetic signals continuously. Every cell produces electrical activity. And where there is electrical activity, there is an electromagnetic field. In other terms, electromagnetic signals are simply energy carrying information, and biology is full of them. But if information can travel through electromagnetic signals in technology, it becomes very interesting to explore how this applies to biological systems. Now, let's look at something we all use every day: Wi-Fi, television, radio, and even car keys. All of these technologies rely on electromagnetic signals, but we don't see them visually, but we fully trust them. Information is transmitted through space without any visible connection, and yet it works perfectly. For example, when you press the button on your car key from a distance, a signal is sent, received, and translated into an action. And this interacts between three fields, which are very particular and which are interacting very often in our everyday life: information, energy, and matter. So the real question is not whether this type of communication exists, because you are using energy, information, energy, and matter even when you press the button of your key. But the question is whether similar mechanisms could also be present in biological systems. In other terms, if technology uses electromagnetic signals to transmit information, why wouldn't biology do the same? We already trust invisible signals in technology. The question is, why would we not trust biology working in a similar way? And based on these principles, we can now introduce the concept of biological resonance. Biological resonance refers to the ability of biological systems to respond to specific frequencies or signals. In simple terms, living systems can interact with specific frequencies. This is very consistent, by the way, with what we discussed earlier: communication, interconnection, and electromagnetic signaling. Some well-known technologies like MRI already rely on electromagnetic interactions with the body. Other systems, such as bioresonance devices or quantum-based approaches, aim to explore these interactions in different ways. For example, devices using broad-spectrum signal emission, such as what you see on the slide now, Tesla-type coils, are designed to interact with a wide range of frequencies. But the idea is not to replace conventional approaches, but to explore additional layers of biological interaction. If biological systems are sensitive to signals, then it naturally becomes relevant to study how these signals can be measured, interpreted, and potentially modulated. Biological resonance suggests that living systems can interact with specific signals, and that this interaction may be measurable and interpretable. Let's now look at a very interesting biological structure, the hair follicle. The hair follicle is a living structure that reflects ongoing biological activity. In other terms, unlike the hair shaft itself, the follicle is a living tissue connected to metabolic and cellular processes. And the hair shaft is mainly composed of keratin and melanin. It is biologically inactive. The hair follicle, on the other hand, is a living tissue. It is metabolically active and biologically responsive. So when we talk about biological information, we are not referring to dead material, but to living, dynamic, and interactive structures. So biological systems often exhibit oscillatory behavior, meaning they function in cycles and rhythms. So these oscillations can be described as sinusoidal patterns repeating waves of activity over time. In other words, biology is not static, it is rhythmic. Life, my dears, is not linear, it is oscillatory. Think about the heart, for example. This is consistent, moreover, with everything we discussed earlier. Nonlinear system, communication, and dynamic interaction. And this is precisely why biological markers need to be interpreted in a functional and dynamic way. The hair follicle is a living biological marker, and biological systems function through rhythmic oscillatory patterns. Let's now look at how the data is processed. The key idea here is that the signals we collect are not interpreted in isolation. They are correlated with large biological databases. This means that the data is compared with reference patterns derived from existing biological knowledge. In other terms, with Epixlife, we are not guessing, we are comparing. For example, one of the main reference centers is located in Hamburg, Germany. But this is not limited to one location. The systems integrate data from multiple international databases, including Germany, Russia, and last but not the least, the United States. This creates a global reference framework. So the key idea is that what we are working with is not a local interpretation, but a globally informed compared analysis. The strength of the system lies in its ability to compare individual data with large-scale biological references. And this is what allows us to move from raw signals to meaningful interpretation. Now, let's look at a real case, anonymized for confidentiality. First of all, you see that we have a case where we observed chronic fatigue syndrome, inflammation, imbalances in the microbiota, and stress. And this example illustrates how different functional imbalances can be interconnected. In this case, several key patterns were identified. As regards the chronic fatigue, for example, the patient presented with persistent fatigue, not just temporary tiredness, but a chronic lack of energy. As regards the microbiota imbalance, there was also a clear imbalance in the microbiota, which can affect digestion, immunity, and overall metabolic function. Signs of low-grade inflammation were identified, which often plays a central role in many chronic conditions. And finally, the last but not the least, significant stress, both physiological and emotional, was part of the overall picture. And you see, they are not isolated issues. They are parts of the same system. So stress can affect the microbiota. The microbiota can influence inflammation. Inflammation can contribute to fatigue. Everything is interconnected. And there is a problem. A purely quantitative approach might look at each parameter separately. As qualitative approach allows us to understand the relationship between them. We are just not identifying problems. We are understanding the system in the whole, like holos in the Greek root, which means the whole. And this understanding is what allows us to define a more targeted and coherent strategy. This case shows that symptoms are not isolated. They are part of a complex interconnected system. And you see, what is very interesting in this case is that the system shows a high need for optimization. This means that several regulatory mechanisms are not functioning optimally. The body is not in balance. It is trying to adapt, but it is still under stress. At this stage, we are not looking at disease itself. We are looking at dysfunction. At this stage, we are not looking at disease. We are looking at dysfunction. And this is exactly where early intervention becomes essential. After targeted interventions, we observe a significantly lower need for optimization. This indicates that the system is regaining coherence and balance, not because we corrected a single parameter, but because we supported the system globally. In other terms, we did not fight the symptoms, but we restored the system. And this brings us to the most important part, intervention and outcome. The intervention combined advanced analysis tools such as BioStar and HeartReader. These technologies allow us to monitor the functional state of the organism in real time. And this is, by the way, what I did to analyze the results of Epixlife and to compare them with my own tools at the beginning of our cooperation. And the results is a progressive and continuous improvement, not a sudden change, but a sustainable evolution. Health is not a switch, it is a process. And to understand this process, we need to look at the modulators involved. This slide illustrates the different types of modulator involved in the system, like microbiome, vitamins, minerals, amino acids, fatty acids, antioxidants, toxic exposure, which is more and more prevalent nowadays, and environmental factors like the food you eat, the type of water you drink, the kind of air you breathe, the electromagnetic pollution, and so on and so forth. And all these factors interact continuously and influence biological function. And this is exactly what we call epigenetic modulation. In other terms, we are not treating the body. We are not treating diseases. We are influencing its environment. And to better understand the patients, we also integrate clinical information. We also integrate clinical questionnaires to correlate biological data with the patient's lived experience. As highlighted by Bruce Lipton, gene expression is influenced by environment and perception. Later on, I will describe you a couple of experiences, if we still have time, performed by Dr. Bruce Lipton in terms of looking at transgenealogical processes and how epigenetic can be inserted into that process. Anyway, I highly recommend to everybody interested into this field to read this absolutely phenomenal book, The Biology of Belief, and see how people's belief can even influence their physical statements. This gave also birth to some collateral disciplines like biological decoding, for example, or psychoneuroimmunology. Whether we fully agree with all interpretation or not, the essential idea remains valid. Biology is dynamic and responsive, and the patient is not just biology. The patient is a complex system of interaction. So what does it mean exactly for the future? The future of biotechnology is moving towards three major directions. First of all, understanding the body as an information system. This is what we call informational biology, but this is not all. Now, if we are looking at the preventive strategies, the key word is acting before disease appears, because you have to remember that when the disease is already present in your body, It means that there is a sequence, a lot of sequences, which were happening prior the disease appeared physically, that might have been understood before in order maybe to prevent the disease to manifest materialistically in the body. And this is where it's absolutely interesting to have this kind of approach. And as regards the personalized approaches, we have to make an adapting intervention to each individual. In other terms, to go from a reactive medicine to predictive and personalized health. In other terms, and finally, the genome is not a fixed destiny. It's a dynamic potential shaped by its environment. And when I talk about environment, I'm not only talking about the external environment. I'm also talking about the internal environment, as you could see also throughout this webinar, that internal environment is as important as external one when you talk and you refer, for example, to emotions. Psycho-emotional systems are playing such a role for the overall health activity of the patients. And there are many other things. I was just referencing previously just an example that I would like to share with you in conclusion. This example comes from Bruce Lipton. They've made a clinical survey of two distinct family, which has nothing to do with one each other. And they studied them on 5 generations. And one of the family had a gene, which is a dominant gene, where there was a marker for a certain type of cancer. And the other family, observed over 5 generations, did not have this marker of gene. But for any reason, the child of the family, which was without marker for cancer genetically for 5 generations, had to be adopted by the 5th generation of the family, which had this marker, this cancer marker. And in about 3 or 4 months' time, we found the same marker, genetic marker, in the DNA of this child from the other family who did not have it for 5 generations. And this is how potent is the epigenetic manifestation. And that's the reason why it's so important to take it into consideration, because this is where biophysics, epigenetics, and technology meet to open new perspective in health, not to replace medicine, but to expand our understanding of life. Muchas gracias. Thank you very much. To conclude, I would say this. Health is not only a matter of structure, it is also a matter of communication, regulation, and coherence. The genome is not your prison. It is a potential. And if that potential can be influenced by environment, signals, and biological interactions, then new perspectives become possible, not only for analysis, but also for prevention and personalized support. This is why I believe biophysics and epigenetics deserve serious attention, because they may help us understand life not as something static, but as something dynamic, adaptive, and deeply interconnected. We do not change life by isolating it. We understand it by listening to its interactions. The future of health will not only be more precise, it will also have to be more integrative. Thank you very much for your attention. And I just would like to add that I thank you, Esteban, very much for having opened this possibility for me to be part of this program, to teach whatever can be teachable, and most of all for the high level of confidence that you dedicate to me, and for which I'm absolutely not insensible as you can imagine. And I hope that this first one will be the occasion of deepening our cooperation in the future. And I would like really to thank you very much and to thank Epixlife and all its members for this new presence that you are delivering to the humanity. Thank you for that.
Thank you very much, Jacques. It is an honor and a pleasure to have been able to share this information with someone of this caliber. A person, a scientist who has worked with James Watson, who along with Francis Collins has been the fathers of the Human Genome Project. A person who has worked with Bruce Lipton, a scientist who has been appointed as a ministerial delegate, as an advisor in preventive medicine, integrative wellness in the community of Savoi, the country of Savoi. I mean, this is the first time in the history of humanity that such a position has appeared. So for us, Jacques, it is a real pleasure to have listened to you, a privilege. We thank you on my behalf, on behalf of Epixlife, for dedicating this time and knowledge to us. And we are very, very grateful. Thank you very much. And a big heartfelt hug from Epixlife. Thank you, Jacques. Thank you all. Thank you, Jacques.